today the topic is on the information argument, aka intelligent design theory. And this is who we are arguing against.
Welcome home. Home? Don't you recognize your old stomping grounds? This is Earth, France, about, 3.5 billion years ago, give or take an eon or two. Smells awful, doesn't it? All that sulfur and volcanic ash. Really must speak to the maid.
Q, is there any point to all this?
Come here. There's something I want to show you. You see this? This is you. I'm serious. Right here. Life is about to form on this planet for the very first time. A group of amino acids are about to combine to form the first protein. The building blocks of what you call life. Strange, isn't it? Everything you know, your entire civilization, it all begins right here in this little pond of goo. Appropriate somehow, isn't it? Too bad you didn't bring your microscope. It's really quite fascinating. Oh, look. There they go. The amino acids are moving closer and closer and closer. Oh, nothing happened.
Now he's very confused at why nothing happened, and that's exactly what we should expect is the point of this talk. I managed to find a perfect frame for the ending there. This video was edited a little bit from the actual one, so just disclaimer there. So here is an example of what Q calls goo. This is the walking protein called kinesin. Short little video there. It's dragging a vesicle along a microtubule. Vesicle is a large thing with plasma membrane with a bunch of stuff in it. It has to go from one place in the cell to another. And the microtubule is like a highway in the cell that the protein walks on. So like this protein is a machine, okay? It's not goo. And it's even more complicated than that because here is what actually the microtubules look like as opposed to that video.
Includes a network of microtubules created by the lateral association of protofilaments, formed by the polymerization of tubulin dimers. While the plus ends of some microtubules extend toward the plasma membrane, proteins stabilize the curved conformation of protofilaments from other microtubules, causing their rapid plus end depolymerization.
Okay, so this is happening all the time in your cells in the positive direction of building a microtubule in the right place at the right time from the right point A to the right point B so that the kinesin can drag stuff along it and other things can happen. And then it's immediately deconstructing at the right time and then reforming again all the time. So if you saw this in a sci-fi show like Star Trek, what would you think? We think this is alien technology. This is nanotechnology far beyond what humans can do. And it's certainly the result of an intelligence. So people have noticed this for a long time. And really, the modern version of the design argument starts really with William Paley in 1802, where he said, if you found a watch in the heath and you didn't know what it was just out there, and you looked at the watch and you looked at all the gears and the way things work inside it, you would assume that it was designed by an intelligence. Okay, so this is the famous watchmaker argument. And then Darwin comes along. I really should have put Darwin on here. In 1838, he comes up with a theory of natural selection acting on random variation and publishes 20 years later in 1859. This is something that Richard Dawkins, a famous atheist biologist, later said defeated Paley's watchmaker argument. We'll talk about that in a second. But moving on with the very first real history of the modern argument from information, teleological argument, starts with a guy named Claude Shannon in 1948, and really before that. And I think he wrote his master's thesis in 1937, which is often called the most important master's thesis of all time, because he basically invented digital computing in it. So he applied Boolean algebra to electrical circuits. And there's a lot of people here who probably know more about that than I do, so I don't want to say too much about it. And of course, he invented information theory to go along with that. And modern intelligent design theory is kind of based on Shannon and information theory. So we're going to talk a lot about that tonight. In 1953, just 5 years after Shannon created information theory, Watson and Crick, pictured on the top right, discovered the structure of DNA was a double helix, and it immediately suggested that DNA was like a digital code. Okay, so here you have a simplified look at DNA. This is the actual structure there they came up with, if you can see it, a double helix. But if you see, there's 4 letters in DNA, A, T, C, G, and they get translated into amino acids. Remember what Q said? The amino acids are getting closer and closer, and they're going to form the first protein, right? These are the amino acids he was talking about. So DNA, transcribed messenger RNA, is translated into amino acids in a long chain, okay? And that suggests a code, a sequence that has information in it, just like a written language, like the words on this page. 1958, Crick gives the sequence hypothesis, saying that, well, what if the hereditary information that transmits information from the parents to their offspring must be in DNA, and it must be in the sequence of bases in DNA. That's the sequence hypothesis. And this is the central dogma of biology. DNA to RNA to protein. If you study biology, you've heard of the central dogma, okay? And I put this here because DNA replicates to DNA and passes it on, okay? So DNA is an information carrier, is what was discovered right around the time we were starting to invent computers and modern computing, which is about digital information. So I thought I'd ask you guys, how would you describe the age of information, which we're in now, not rhetorical? Why is it called the age of information?
Seems like information is just like at our fingertips.
All the time, yeah.
Easily accessible.
It's like that's not my phone. I can look up anything I want on the little gadget here, and it's like, oh, information is right at my fingertips. The internet, right, is a highway, information superhighway. It used to be called that when I was growing up. No one uses that word anymore. But it used to be called the information superhighway. So what is information in theory, right? Shannon said that information is a measure of uncertainty. And that is somewhat counterintuitive, because when we think about information colloquially, like just you and us talking, I gave you information, we would think that, well, if I get information, I get more certainty of knowledge, right? But Shannon counterintuitively suggested that actually when we're dealing with digital information in a message string, information is better understood as a measure of uncertainty, okay? And so the tricky part about understanding information theory is understanding why we think of it as a measure of uncertainty rather than certainty. Information at its most fundamental is about narrowing possibilities and thus about reducing uncertainty. The more uncertainty is reduced, the greater the information. So even when we talk about more information, the way we talk about it is we're reducing uncertainty rather than increasing certainty. And there's a reason for that. This is a quote from William Dembski. Another quote from William Dembski: information is the costs required to raise a smaller probability to a larger one. So if I want to increase the probability that I will get a job as a lawyer, I want to gain information about the law by going to law school, right? And that raises the probability that I can become a lawyer or something like that, right? So when you gain information, it increases the probability that your target or your desire or your information is correct or will happen. Now we need to talk about digital versus analog. Okay, so I got these paintings from Adam from last month. This is Jeremy Gooding's awesome painting on the left that requires skill. And here's the painting on the right that does not do any of those things. But the information in the paintings is analog information. Okay, that means it's like a curve, like a mathematical curve or something like that. And it requires, if you want to copy a painting like this, it requires a lot of skill. It requires you to exactly copy the lines in the painting and the colors and everything else. Whereas digital information is just whatever, man. Like if you want to copy this line, digital versus analog, you can do it this way, you can do it this way, you can write it in handwriting and block letters, you can use magazine letters and use it like a ransom note. But it's still the same information, right? Because it's still the same letters using the same code, okay? So digital information is special compared to analog information because it is based on a code. And when you copy it, you don't need to be an expert artist. Any of you could copy this information here. None of you could copy this information. Maybe that information you could copy, which is a really good argument against modern art, by the way, which again, we talked about the first 2 months. Information is immaterial because it is independent of the medium. So one medium would be a television, right? Another medium would be a laptop. Another medium would be a paper that I write it on. Another medium might be radio frequencies or the internet, Wi-Fi, or just about anything. Your brains can carry information. What happens when, say, this phrase, digital versus analog, is when you take that information, you copy it on a piece of paper? Where does it go? When the information leaves the screen here and goes to the paper that you copy on it, is that a physical transfer? I don't think so. That's an immaterial transfer of information. There's nothing physical that transfers. Now, there may be some materialists that would dispute that, but when you copy it, you can do it any different way you want, and it's still the same information. If it's digital information, it is independent of the medium. It doesn't matter what medium you use, it's still the same information. Okay, so I tried this explanation out on Clark earlier, and I saw a light bulb go on, so I thought I'd put this in my presentation. These are the difference between FM and AM radio, right? So frequency modulation transmits information from one point to another by increasing or decreasing the frequency of the periods or the wavelength, or right, I guess the periods. And then amplitude modulation increases or decreases the amplitude, makes it smaller amplitude or higher. And so what you need to transmit information this way, like if you saw this show up, right, you'd have no idea what it means unless you had a code to decode the information, okay? So when you transmit information from point A to point B, and we're really talking about where information theory came from here, literally, like this is what they were doing. Shannon was a code breaker in World War II. And the Allies famously broke most of the German codes and the Japanese codes in World War II. And so they were dealing with this in a wartime situation. So they're decoding the codes that they were using to transmit information, okay? And so one thing that you want to note, right, is that, and this is really the main reason why information is a measure of uncertainty, is that the same amount of information in the sense of the same number of wavelengths of your radio signal can communicate a whole lot of information or less information based on the complexity of the code that you're using. Okay, and the reason for that is that if I have a code that's very simple and I have a lot of this same radio signal, right? When we receive the signal, you get a very simple message, right? But if the code is much more complex, nothing changes physically about the radio signal. You've got the same signal as long as the code is right. But if the code has many, many possibilities, like millions of possible outcomes of the code, or possible things the code could be, you're excluding way more possibilities with the same exact radio signal. And when you exclude more possibilities, you have more information. And this is actually kind of how some versions of data compression work, where you increase the complexity of the code, and that allows you to transmit more information in the same physical medium. Okay, so I brought an illustration of this since I'm a millennial. Okay, this is what I used to carry my CDs around in, my music CDs. And if this thing was full, it'd be bulging, right? So I took this everywhere when I went on long drives so I could listen to all my music, right? Now, I can put it all in my phone. Or I don't even put it on my phone anymore, I put it on the cloud, right? Because the physical medium in which it's in is now an MP3 or something like that, which is you can have more information in less space because of the code that's being used is more complex, basically. Again, I know there's computer people here who are going to be like, but we can talk about that later if you want. But increasing the complexity of the code increases the total number of possibilities that could be transmitted by the same code. And that means any single message string communicates more information. Why? Because it excludes more possibilities. Okay? And so it's actually the number of possibilities that dictate how much information you can have in a message string, which is a measure of uncertainty. The whole range of possibilities is uncertain when you have just that, right? So how many possibilities are you excluding determines how much information you're passing on. Okay? All right, so a simple explanation of this, I'll try to run through this quickly, is which of these pictures has more information in it? Obviously the bottom, right? So the question is kind of why? Okay. If I have 100 kinds of dogs, let's stipulate that's just how many there are, and I tell you that I have a dog, how much information am I giving you? Well, it could be any one of the 100, right? So I'm giving you a possible information that says I have any one of the 100 dogs out of the 100 that there are. So that's a really low amount of information. But if I say I have a lab, let's say 10 of the 100 kinds of dogs, so we're talking about, this is the code now. 10 of 100 kinds of dogs gives you more information because that excludes 90 of the kinds that it could have been, right? And so that, if that's 10 out of 100, that gives you 0.1. Well, the problem is 1 is greater than 0.1. So, but we know that bottom one is more information than this, right? So what we do is we take the reciprocal, okay? Okay, so that's what I'm doing here. I'm just taking 100 over 10 to give a sort of ad hoc information measure, okay? As opposed, I see Harrison nodding, he's a math guy. I hope everyone else gets this. So if I say I have a black lab now, that's 1 out of 100, and that says, okay, I gave you more information now, and so now we've got the reciprocal of 1 out of 100, and that gives you 100, which is more than 10, which is more than 1, okay? So we take the inverse of an uncertainty measure, meaning the uncertainty measure is the size of the code, right? And the message string is the information you actually have, okay? But the measure here, this number, is really a measure of the size of the code more so than the measure of the information you have, because if you want, you can increase the complexity of the code and say, okay, there's actually 1,000 kind of dogs. And then if I tell you I have a black lab, right, I'm actually giving you 1,000 information instead of 100. And nothing changed in what I said except the size or complexity of the code. So this is, we don't need to talk about this too much. This is just what bits are, is binary information digits. And so this is the simplest kind of code that could exist is ones and zeros, right? It's a binary code. And so you can see even a string of 100 places long gets you a number of possibilities that are 1 times 10 to the 30th. So even a very short code with a very simple code with a short string can get you a lot of possibilities really quickly. Okay. DNA is also a digital code, and it's a 4-character code, A, C, A, T, C, and G. Okay. And in proteins, it's a 20-character code for 20 amino acids. Okay. And so you can imagine how many possibilities there are in such a code when the string gets long. Okay. This is the sort of beginning of the modern intelligent design movement. We've got a book called Mystery of Life's Origin by Charles Thaxton, Walter Bradley, who just died recently, and Roger Olson. Evolution, a Theory in Crisis by Michael Denton was an important book. Michael Denton is a biochemist. And then, I forgot, you have Blind Watchmaker by Dawkins, which sets off a whole discussion about this me thinks it is like a weasel program. Okay, so Dawkins suggested that evolution, of course he's an evolutionist, and so he's trying to explain how you can get protein sequences, DNA sequences that actually contain information out of a random starting sequence, okay? So he wrote this simple program that just changes one letter every time. So this is 10 iterations, this is 20, 30, 40, 50, 60. And each time, right, the program checks to see if it's the right letter in every position, and then locks it in if it is, and then allows it to vary if it's not. And eventually in 64 iterations, so Dawkins imagines this as being 64 reproductions in biology, like 64 new creatures, or new genomes really. In only 64 generations, you can get me thinks it is like a weasel out of it. Now, eventually, Dawkins realized or admitted that this is not how biology works. And famously, Winston Ewart was one of Dembski and Robert Marx's students, and this is what got him going into intelligent design also, because he was a big computer programmer. And he's like, wait a minute, right? The reason that Dawkins's program works is because the target phrase, the target sequence is already in the program. Okay, it's in the program, and the program is always checking the string that exists against the target sequence and accepting it if it's like the target sequence and rejecting it if it's not. But biology doesn't have a target sequence. Okay, there's nothing in biology that has the information that's supposed to be in DNA already there while the real DNA like measures itself against it, right? Unless you're talking about some kind of God. Okay, so a big history of debate around this kind of, we're going to talk about that later. Some more history of effective intelligent design. Edwards versus Aguillard was a Supreme Court case that supposedly ruled you can't teach creationism in schools, public schools. Of Pandas and People was written in 1989. This became an issue in 2005 in another court case. 1990, Discovery Institute is founded, which became the primary institutional home of intelligent design. 1996, Irreducible Complexity, an idea by another biochemist named Michael Behe. It's published in this book called Darwin's Black Box. I should have brought my copy of it. I read this book in high school, I think in 1998, and I studied biochemistry in college because of it. Very interesting book, still holds up to this day. 1998, The Design Inference is written by William Dembski, and he outlines the idea of specified complexity, and we're going to spend most of our time talking about that. 1999, Dembski goes to Baylor, a supposedly evangelical school, with Robert Sloan as the president, and starts the Michael Polanyi Center. Polanyi was a mathematician, scientist in the mid-20th century. Dembski is outvoted apparently by the faculty at Baylor 27-2 to remove the Michael Polanyi Center. So right from the start, intelligent design is being discriminated against in the academy, most likely because they're afraid about getting their federal research grants revoked. Certain features, so this is what they always said for a long time, this is kind of the boilerplate. What is intelligent design theory? just says that certain features of the universe and of living things are best explained by an intelligent cause. Okay, but it's quite a bit more complicated when you make the argument formally. It is a scientific theory that does not reference the Bible, and it takes no position on natural history. So they don't care about the age of the earth, they don't care about whether we actually descended from a universal common ancestor. So some intelligent design theorists accept common descent, like Michael Behe is an example, meaning we all descended from a single common ancestral population. All that intelligent design theory rejects is the mechanism of neo-Darwinian evolution, which is selection acting on mutation. Okay? They say this is what cannot work. It cannot produce new information, this mechanism. Okay? Only an intelligence can produce this kind of information that we find in DNA. Okay, so what is specified complexity, Dembski's general formulation of the design argument? Well, suppose we're playing cards and I get a royal flush, and not only do I get a royal flush, but I get it 4 times in a row. Are you going to accuse me of cheating? I would hope so, because you guys are smart. The probability of a royal flush in spades, let's say I get a royal flush to the ace in spades 4 times in a row. How about that? I'm cheating? I think so. The probability is 1 in 2.6 million, okay? But because I'm a clever fellow, I will argue that actually every one of the hands that you guys got, that's the same probability. Your hand of a 3 of hearts and 5 of diamonds and blah, 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 also has the same probability. So what's the problem, guys? My hand had the same probability as your hands every single time. In fact, every hand has the same probability. So you can't just say that I'm a cheater because my hand was improbable. Okay? Neener, neener, neener. I already did all that. So why do we still conclude design in this case? We're talking about cheating, right? Fixing the deck or stacking the deck. Well, Dembski suggested that the reason we still conclude design in this case is because there's an additional component of a design inference called a specification. It is not just high probability or low probability, which is what he means by complexity. Remember when we were talking about a message string? The high complexity of the message string is the number of possibilities in the code, right? So when we say complexity, we're really just talking about the number of possibilities in a code like that. So something that has high complexity, and if you've only got one message string, it's a low probable result, right? Where if it's a very simple code with very low number of possibilities, it's low complexity or high probability. So Dembski comes up with the explanatory filter to explain all this. I'm going to move on to the universal probability bound. Well, let's go through it real quick. So he suggests that when we infer design, you start up here with our designed or potentially designed thing, and we go, is there contingency in it? Meaning, could it have been otherwise? Okay. If no, then we say the cause of it is necessity. It must have been that way. In all possible worlds, there's no other possible result because there's no contingency. So we explain it by reference to necessity or law, like scientific law is like necessity. If there's contingency, we go, does it have complexity or low probability? If no, we say, well, it could have just happened by chance because it's just highly probable. It's not necessary, but it's very probable that it would have happened on its own. So we say chance is a good explanation. But if it's very high complexity, we say, yes, it has complexity. And then we ask if it has a specification. We'll talk more about what a specification is later. If it has a specification, then we're justified in inferring design. Okay, but if it doesn't, we also go to chance. The universal probability bound is just a kind of a back of the envelope calculation of how many possible events that could have occurred in the history of the universe, right? So we've got 10 to the 80 atoms. I think that's actually the mass of the universe. 10 to the 65th, I think, is the number of actual atoms. But we're being generous, okay? So 10 to the 45th, I think, is the rate of the fluctuations in a cesium nuclei, which is used to regulate atomic clocks, okay? So it's one of the faster processes we know. 10 to the 45th rate of events per second. 10 to the 25th number of seconds, and that gives you a universal probability bound. 10 to the 150th, the total number of possible events that could have occurred in the entire history of the universe. And this is on the old Earth scale, right? So I think they use 10 to the 10th years, which 10 to the 10th is not a huge number when you're dealing with 10 to the 150th. It doesn't actually matter that much. Right, which is a feature of this argument. It actually doesn't matter much how much time you have to evolve something when you're dealing with numbers this big. Another probability bound or something like this, we'll talk more about the biological probability bound later, but basically 10 to the 30th is the estimated total number of genomes that are produced on Earth every year, and the vast majority of this is bacteria, okay? And so 10 to the 10th is just 10 billion years. And then 10 to the minus third is the fastest rate at which mutations occur in bacteria. So 1 out of every 1,000 in fast mutator E. coli's genomes have a mutation. For humans, it's a higher rate. It's about 100 per person. But humans are negligible biomass compared to bacteria to this 10 to the 30th number. So we just say 10 to the minus 3, and Behe was generous and said, we'll pretend that it's actually one mutation per genome. That's the rate of HIV about. And compare that to the Powerball. Your odds of winning the Powerball are 1 in about 300 million, or roughly 10 to the 9th. Okay, so we're talking about massive numbers, even on the biological probability bound. Okay, so if complexity is a raw measure of the total number of possibilities and a specification, we need to talk about a specification. So what is a specification? Well, originally Dembski described it as an independent pattern, meaning independent of the thing which gives rise to the complexity of the object. So a good analogy here is the archer-target analogy. So we know instinctively there's a big difference between an archer who fires at a target and hits the bullseye and an archer who fires at the broadside of a barn and then paints the target around wherever the arrow hit, okay? So the pattern of the target has to be independent of the process by which the arrow got there, okay? In order for it to be a valid specification, okay? Because otherwise it's cheating, right? We're just making things up. Okay, but now he's decided that this concept is just included in a description of information that's similar to something called Kolmogorov complexity, which is basically just the shortest algorithm that can reproduce some message string. Okay, but he's simplified it and he just says, look, a specification is a short description of something. So when we say something like a universe supporting complex life, that's 4 words, and there are about 200,000 used often words in the English language. So when we use a 4-word combination like that, we're giving some information, right? Because we're giving a 4-word string in a code that has 200,000 possibilities. Okay, so he's saying a short description is different than a high complexity. And when you compare the two together and put them together in an equation, he has a whole technical defense of this in the 2nd edition of the Design Inference, which just came out a few years ago. We have something called complex specified information. If both high complexity or low probability and a specification are present in some message string, we are justified in inferring design. And Stephen Meyer extended this into an inference to the best explanation, which is how scientific reasoning works. And the best explanation for the existence of specified complexity is intelligence, because it's the only thing that we observe creating specified complexity is intelligence. All right, so here's some examples to help you guys think about this in case I was talking too fast. Vortices and crystals are an example, like snowflake here, are an example of a specification, right? Because we can see a clear pattern and we can give this a short description. This is a snowflake and it's a crystal pattern or something like that, right? It's a specification, but it has low complexity. Why? Because we know that snowflakes are produced by natural laws that form these patterns. And natural law is an example of necessity, meaning it's going to happen a certain way, okay? So a design, we might think intuitively that a snowflake is designed and we might believe it's designed, but on the design inference reasoning, it's not going to trigger a design inference. Doesn't mean it's not designed, it just means it doesn't trigger a design inference according to specified complexity. The opposite example is like sand on a seashore. There are, just millions, trillions of grains of sand there, and the specific arrangement of the sand grains is highly improbable. Any arrangement of the sand is highly improbable. So that's a very high complexity. But there's no specification, right? I look at that and I'm just like, there's no message there, there's no information there, it's just there. And there's no rhyme or reason to the organization of the sand on the seashore. So this also does not trigger a design inference. But Mount Rushmore is an example of both. We have this huge mountain, right? There's all kinds of rocks there and particles in the rocks that are arranged a certain way. It is very improbable, but that fits a specification. We can say, oh, that's George Washington. Short description. That's, okay, that's TR, that's Lincoln, and this is Jefferson. Is that right? I couldn't remember who that was. So Mount Rushmore is clearly an example of where we would infer design by this criteria and also just intuitively. Okay, so going back to biology now, the Human Genome Project was sort of completed in 2003. It's a whole nother story there. In 2004, a scientist named Douglas Axe completes a study of the probability of protein folds, or it's called estimating the prevalence of protein folds or something like that. I look up this paper all the time and I just type in estimating the prevalence, so I don't remember the rest of the title. What Axe's study did was show that something called protein folds, which is how proteins fold into a specific shape that helps them perform their function, are highly improbable in the sequence of amino acids. So remember, we have proteins are a sequence of amino acids, right? The amino acids are getting closer, where they bond into a long string of amino acids, and the number of them is very high. But the number of those sequences that will fold into a stable protein, a functional protein that's stable, is very low. And I'll show you more about that in a second. Stephen Meyer publishes on this, extrapolating Axe's work into an inference to the best explanation. And he immediately draws controversy because he published it in a Smithsonian-based journal. The editor there was an evolutionist at the time, and he was forced out of his position. There ended up being 2 congressional hearings about this that determined that Sternberg was treated unjustly just because he published Meyer's paper. In 2005, there's this court case in Kitzmiller versus Dover in Pennsylvania, where that book Of Pandas and People, they put it in the library of the school, and then they wanted to put a sticker in the biology textbook that told students, this book Of Pandas and People is in the library if you want to check it out. And so this went to court, and they were ruled against that you can't do that. Most of the design proponents were against this at the time, but they got called in as expert witnesses in the court case. Many people, even today, who are skeptics of intelligent design or just atheists in general, will say that intelligent design was dealt a death blow in 2005 by this court case, as though court cases decide scientific issues. In 2007, the ENCODE study was published. The ENCODE study showed that upwards of 80% of our genomes are being transcribed. And the evolutionary prediction for many, many years, well documented, was that the vast majority of the genetic code is not doing anything. It's junk DNA. It's leftover detritus, debris, junk, trash from the evolutionary process that is random and has no direction or target sequence, like the me thinks it is like a weasel. Okay. But ENCODE showed that why is that your body transcribing almost more than 80% of your DNA if it's not doing anything? That doesn't make sense. So this was good evidence that design theorists were on the right track, and design theorists and creationists alike did predict that this would end up being the case, that we would find that this junk DNA actually is doing something. Dembski and a professor at Baylor named Robert Marks started the Evolutionary Informatics Lab in 2007. This is shortly after, I think, Dembski and Sloan both left. But Marks kept going because he had tenure, okay? And Winston Ewart studied there. There was still controversy over it. 2007, his second book is published. This is a much more complex and interesting argument that I don't have time to talk about. And Signature in the Cell was the big book by Meyer, where he takes his paper from 2004 and puts it into a book form. Okay. And by this time, intelligent design, in my opinion, has essentially won the intellectual argument. Okay. So let's talk about Axe's work and why Axe's work is important. This is the protein hemoglobin. It has 4 subunits. You can see the 4 different colors, and they're all connected, okay? And what it does is it binds oxygen, okay? And all your proteins and all the functions in your body, almost all of them are performed by proteins just like this. This is a representative, these are all individual like atoms, right? And there's a long amino acid sequence, okay? This is called the primary structure, is the sequence of amino acids all in a row. Here's a secondary structure where it coils into an alpha helix, right? So that's just the same long chain coiling into an alpha helix. Here's another one called a beta sheet where it kind of goes back and forth like this. And then it folds into tertiary and quaternary structures that actually perform a function like a machine, okay? Axe studied a protein called beta-lactamase, and he specifically studied 153 amino acid folding domain of a beta-lactamase called TEM1. If any of you have done an experiment in a biology lab where you used ampicillin as a drug, okay, lactamase is used because, oh shoot, I should have reviewed this, but it's sensitive to ampicillin. And so you can test and see whether the sequence got into the bacteria by whether the bacteria lives or dies based on when it grows in an ampicillin medium. So I think its lactamase can digest ampicillin. So if it has beta-lactamase, it can live even in the presence of this antibiotic, okay? So it's a very, very commonly used lab enzyme, protein. Okay, so that's kind of the raw complexity measure of this folding domain of TEM1. Just big numbers, really impressive. You're supposed to be like, whoa. Okay, Axe found the probability of stable functional folds to be 1 in 10 to the 77th power. What this means is that the number of stable functional folds that actually perform the function of beta-lactamase are only found in 1 in every 10 to the 77th sequences, okay? Again, comparable to the mass of the entire universe at 10 to the 80th. Okay, so this is literally like finding a stable functional fold that does what beta-lactamase does, or at least what this folding domain does, is like telling somebody, okay, I'm thinking of a single proton or neutron in the entire universe. Okay, I know which one it is. You've got 1 chance to go find it and pick it out, okay? That's not going to happen, okay? Axe just basically extrapolated this in his original 2004 paper to all proteins of the same sequence length, okay? So any protein of 153 would be about 1 in 10 to the 74th probability. Because he's just assuming that, he's just saying there are about 1,000 proteins of that specific sequence length. Okay, so he just multiplies it by 1,000 to get that. Okay, let's skip this. Okay, so we're back at the biological probability bound where we've got about 10 to the 40th genomes per year. being generated, or total, actually, sorry, since the history of life on Earth being generated. Okay, so this is a total number of possible chances that evolution has to find a target sequence. Compare that to 10 to the 74th, okay? And that's just one protein that is 153 amino acids long. And the total number of genomes in the entire history of life just would not be expected to find even a single folding domain of a single protein by a random search. Okay. Compare that to the Powerball that I showed earlier. This is like finding this single stable functional protein fold by random chance is like me winning the Powerball 4 times in a row. Okay, that's the probabilities we're talking about here. Now, a guy named Dan Tofik, I think he was at Hebrew University, I know he was in Israel, did a study in 2006 where he studied 10 different proteins with vastly different structures. He found that 2/3 of all mutations in those proteins reduced the thermodynamic stability of their fold by 1 kilocalorie per mole or more. So a protein is under a lot of pressure thermodynamically, right? It's like a rickety roller coaster. There's a Thunder Wolf. It was like at Worlds of Fun, if you've ever ridden that one. That's an old rickety wooden roller coaster, and it's always shaking, right? That's what's happening to proteins at the molecular level all the time because of thermodynamic forces. So they're constantly being buffeted so that they're going to, if they're not very tightly bound together in their shape, they're going to fall apart, okay, from that. And so that's what thermodynamic stability means. These chains that are folded into these complicated three-dimensional shapes, if they're not stable enough, they're going to fall apart and become just a loose chain in solution, like spaghetti in water, right? And they're not going to do anything. Okay, so if 1/3 of mutations work, because 2/3 do not, this leads to a generalization of X's result. So all we have to do is remember the way we calculate the total number of possibilities is the number of amino acids in the code, which is 20, to the power of the length of the sequence. Okay, so we're taking the number of amino acids at work, which is just 1/3, and we're raising it to the power of the sequence, which in Axe's case, he studied a sequence length of 153, and look at that. You get 10 to the negative 73rd power. Okay, so this is an independent confirmation of Axe's study from a completely different method. Pretty powerful stuff. Scientifically, that's like, whoa, it never happens, right?
If this has been helpful to you, please like this video and subscribe to the channel.